नहीं हो रहा और जरा रखा करना और ये चुप कर ले
Hello, class. Very good evening. Um, I hope you're all joining us soon. A very warm welcome to the third day. Good evening. Very good evening. Very few students are joined us today. Why? Everyone on vacation. It's already 6:05. Now few students are joining us. Mhm. So let everyone join. Then we start with today's lecture. How are you all, students? Okay. So we are a small group, not big. Let's few more students to be there. Then we start our lecture today. 1 minute more. So students, are you all there? You have joined it. I think that's the maximum students are there. Or shall I wait for some time? Tell me with yes or no. Shall we wait or we should start now? It's already, let's start at 10 then, 16:10. If somebody has to join us, they will join us. And how was your day? Busy day? Lot of things going on. So there is second wave coming up of corona. So just be careful. In Spain it started. In Germany it started. They started to have lockdown again. And be careful in your house also. And we're also having winter now. Winter is coming, like Game of Thrones they say. Winter is coming. So we better be careful because in winter we have a lot of cough and all these things. So try to avoid early morning walks, especially in Delhi where we have a lot of pollution. Try to do things when you're going outside to be fully packed from now onwards. And I'm acting like your father or mother now here, but it has to be like this from now onwards. We have to maintain very nice schedule for ourselves to face the winters because the corona wave will be coming again in upcoming time. So we never have thought that we have to see these kind of threats from one virus, but this is how it is now. um, Please, please, please take care of your older persons, whosoever in family older, whosoever have diabetes, sugar, or any heart problems, cardiovascular diseases. They should be really careful from all these things. um, they have to be taken extra care. So please, my request to you all, take care of your family and yourself first also. So on that note, we will start. No, not this one. So qualitative and quantitative traits. Somebody was asking yesterday about them. What are these traits? So we today are gonna deal with these of upon these two traits upon. So let's start from today's lecture from your unit one, topic one. We are done with the Mendel's law yesterday of inheritance. Types of gene actions we are done. So today we will be going with the qualitative and quantitative traits. So what is the difference between qualitative and quantitative actually? So they are both actually a phenotypic traits of different organisms, maybe of two kinds. So phenotypic traits could be of two types, that is qualitative and quantitative. So qualitative traits are classical Mendelian traits such as form, structure, pigments, antigens, antibodies in the humans, and so on. So what are the traits that we have studied so far? So all these traits are coming up in this group as a qualitative traits. So they come under the genetic control of single gene with no or little environmental modification to obscure the gene effects. So environmental effects is very less on these genes. And qualitative traits have distinct phenotypic classes that are said to be discontinuous variations. Okay. So organisms possessing qualitative traits have distinct phenotypic classes and have said to have discontinuous variations, and there is little or no environmental modifications happening on the effects, and they are of three types like structure, pigments, form, antigens, antibodies. So they belong to that part. Then quantitative genetics, according to quantitative genetics, that is inheritance of multiple genes. Um, they are the degrees, phenotypic traits of degrees such as height, weight, skin, yeah, skin pigmentation, susceptibility to the pathological diseases, intelligence in man, amount of flowers, fruits, milk, meat, or egg produced by plants or animals, etc. The quantitative traits are also called as metric traits. So they are in the metrics that we are measuring something. So we are able to measure them. So that's why they come into this. Um, in contrast to qualitative traits, the quantitative traits may be modified by the environmental conditions. So, so environmental conditions could modify them, these quantitative traits, whereas the qualitative traits could not be modified by environmental conditions. And they're usually governed by many factors, each contributing the small amount of phenotypes individuals, such as detected by Mendelian methods or by statistical methods. So genes which are non-allelic and have effect of a single quantitative trait are called polygenes or cumulative genes. So genes which are non-allelic and effect of a single quantitative trait are called polygenes or cumulative genes. So the inheritance of these cumulative genes or polygenes or quantitative traits, all as a same term, is called as quantitative inheritance, multiple factor inheritance, multiple gene inheritance, or polygenic inheritance. All seems to be same. And genetical studies of qualitative traits are called quantitative genetics. Further, if you are studying the qualitative traits, that is, studying of that trait is called as quantitative genetics. So these are some basic terms to identify what is quantitative genetics. That is, they are the one which has affected by the environment. They are metric traits. They are known to be by like height, weight, skin pigmentations, and all these, which could be the, we can measure something that belongs to the quantitative genetics. And study of quantitative traits is called as quantitative genetics, and study of qualitative traits, sorry, qualitative traits is called as qualitative genetics. So certain characteristics of quantitative inheritance, that is, so we discussed some definitions earlier. Now comes some characteristics of the same. So segregation phenomena occurs at indefinitely large number of gene loci. If a substitution of allele occurs in a gene locus, then such allelic substitution have trivial effects. The genes for multiple traits have different biochemical functions, but similar phenotype effect. Therefore, the phenotypic effect of gene solutions are substitutions are interchangeable, and these have pleiotropic effects. That is, one gene may modify or suppress the other phenotypic traits. So like yesterday we discussed epistasis, so one gene could modify the other gene. So the polygenes have these effects, and single allele may do only one thing chemically but ultimately have many characters. And environmentals have a naive considerable effect on the phenotypic expression of polygenes. So for like height is genetically controlled by the quantitative trait, but some environmental factors such as soil, fertility, texture, water, temperature, duration, wavelength of incident lights, or occurrence of parasites could also affect the height. So all these things, they comes into the environment, and they could also affect the quantitative traits. So environment also play a big role in maintaining the quantitative traits. So some examples of quantitative inheritance. So there's a kernel color in wheat. So the seeds that we have, the kernels. So there's one example done by the Nilsson, Abel, and East in 1909 and 1910 and 1916. So they worked together, and gave first significant clue of quantitative inheritance. And the individual works on the wheat. The work was on the wheat actually. So they crossed a strain of red kernel wheat plant with another strain of white kernel, and grain from F1 was uniformly red, but the shade of intermediate between red and white of parental generations. So what they see, they cross a red kernel with white plant uh, or wheat plant with other strain of white kernel. So they crossed red, dark red and white, and in between they saw the F1 generation to be uniformly red. So they saw some medium color, medium red in between. So they must suggest that something incomplete dominance. Like yesterday we discussed about Mirabilis yellapa, the 4 o'clock plant, with the factors of red and white plant. So what we saw was that they were giving pink color. So same way there is same in this case is happening. So we have a mixed color, medium red color. F1 offsprings were crossed among themselves, and F2 zygotes showed 5 different phenotypic classes. So that was something new that we have never discussed them before. So that was happening in this quantitative inheritance. So they gave a ratio of 1:4:6:4:1. So one was dark red in F2 generation, then was the red, then was a medium red, then light red, then white. So like this we have different colors to our work. Yeah, all clear. So noting that 1:16 of the F2 was extreme in color as either of the parental red or white. So 1:16, your dark red and white was the extreme colors of dark red and white, and they theorized the 2 pairs of genes controlling the production of red pigments while operating in this cross. So they were the 2 colors which were mainly playing major role in your giving pigmentation to these different colors in between. So each gene was supposed to contain 2 alleles. So one allele producing given quantity of red pigments, and a counterpart did not produce any pigment. So if we symbolize these genes for red with capital letters A and B, and the alleles resulting of lack of pigments production by small a and small b, we can illustrate the cross as 1:16 red, 4:16 dark red, 6:16 medium red, then 4:16 light red, then 1:16 white. So like this we have uh, cross. So we have AABB, small a, small bb, and then we have the F1 generation to be medium red, and then we take their gametes according to the law of segregation, and then we had again one Punnett scare by having the each types of it, and then we made this cross, right? So we made this cross, and this cross we saw that very nice uh, crossing giving you 1:4:6:4:1 ratio of a red, light red, medium red, light red, and white. Then quantitative genetics, inheritance of multiple genes. Another classical example of polygenic inheritance was given by Davenport in Jamaica. He found that the 2 pairs of genes, capital A, capital B, small b, causing the difference in the seq pigmentations between. Now there comes the next example of Negroes and Caucasians people, like having a black color and the white color. So these genes were found to affect the characters in the additive fashions. So thus, true Negro was 4 dominant genes with this capital A, capital A, capital B, capital B. White has been being recessive genes with small a and small b. So F1 offsprings of mating with small a, small b with capital A, capital B we have a mixture of capital A, small a, capital B, small b. And we intermediate skin color we call as mulatto. We call them as mulatto with the crossing of uh, Caucasians and black Negroes. So mating of such mulattoes and further produces a wide variety of skin colors, offsprings, ranging from dark to original Negro parents to the white as original white parents. And we saw again the same ratio 1:4 is to 6 is to 1. So again we have Negroes parents, then white parents, and then mulatto is the F1 generation. Then comes the uh, mulatto. Then we have these generations like this one, capital A, capital B, capital A, small, capital B, B. So like this. So we have these different ratios. So these are genotypic frequency. 1:2:2:2:4:1:1 2:2:2:1, and this is the together as a phenotypic ratio, as black, dark, intermediate, light, and white. So like this we have uh, uh, the same way that is your quantitatively we saw these data. Then comes the height in men. Skin color in man is rather simple of polygenic inheritance because of two pairs genes are involved. So inheritance of height in man is more complex phenomena involving perhaps 10 or more pairs of genes. So if one the characteristics of tallness is recessive to the shortness. Thus, individual having a genotype of more dominant genes will have the phenotypic of the shortness. So here you can see the height in inches on the x-axis and number of meters on the left on the y-axis. So here we also see the same trend like that when we made a graph in a population from 140 centimeter to 203 centimeter. So there are many examples could be there like the same. Some more examples are there. Now we are done with the quantitative traits. Now comes the next as your qualitative characteristics. So before we go, till now, everything is clear, students? Yeah, they are actually. So I was teaching in the college, so we were supposed to teach you the same syllabus. So I was teaching same in the agriculture university, this principle of genetics. So the syllabus is the same from TNAU notes. So I just transform into presentations and we will continue like that. I hope it will be fine with you students. So now next part, that is qualitative characteristics. The easiest characteristics or traits that deal with those involving discontinuous or qualitative traits which having governed by one or few major genes. So in this case, one or few genes are playing a bit major role in making your quantitative characteristics. So examples are starchy versus sugar kernels, and determinant versus indeterminate habit of growth in green beans. So they are expressed qualitatively. So qualitative traits, they are expressed qualitative, taking the quality, which means phenotypic falls in different categories. So the pattern of inheritance of qualitative trait is typically monogenetic, that there will be single genetic level, which means the trait is only influenced by the single gene, not by major genes. So some major differences between qualitative and quantitative traits. So qualitative genetics and quantitative genetics. So the qualitative genetics, it deals with the inheritance of the trait of kind, like form, structure, color, etc. So it includes the inheritance of the traits of the kind. whereas the quantitative, it deals with inheritance with the traits of the degree, that is heights of length, weight, number. Qualitative genetics deals with the discrete phenotypic classes occurs which display discontinuous variations, and spectrum of phenotypic classes which contains continuous variations. So here we have continuous variations in the quantitative, and on the side on the qualitative it's a discontinuous variations. So here the qualitative traits are governed by two or many alleles of single gene, whereas quantitative traits are governed by polygenes, many genes actually, non-allelic genes. The phenotypic expression of gene is not influenced by environment. So in the qualitative environment is not playing big role, but here, as we have discussed, environment play a major role in this polygenes effectiveness. It concerns with the individuals mating and their progeny, where it's concerns with the population of organism concerning the all possible kinds of matings. Here it's made by the counts and ratios, it is made by the statistical methods. Quantitatives are always by the statistical methods. Here we just counts and we make a ratio in the qualitative traits in the genetics thing. So that's one more example difference between qualitative and quantitative traits. So here you can see qualitative, then quantitative, then there is a threshold. So in the qualitative traits, you have blood, serum types, enzyme, and effects. So mode of inheritance is simple Mendelian, environmental effects is little. So quantitative traits, stature, height, intelligence, milk yield. In the quantitative, we include all these. So mode of inheritance is complex and moderate to great is the environmental effect in this case in the quantitative. So this is the graph of qualitative traits, where we have normal frequency and comparing to the disease frequency. So we're just comparing in the graph way. So there's not so much of statistical data or so. But in the quantitative, you have a very nice bell-shaped curve like this. So this is what we have to discuss in this chapter. So to continue the same thing, I will like to take you with the quantitative traits that we were discussing. So very small chapter, multiple factor hypothesis. So that is also quite nearby. So any question, students, so far? Yeah, qualitative, that's why they are called monogenic or oligogenic, whereas the quantitatives are in polygenic states. So let's continue the next topic with the same thing, that is principle of genetics in the multiple factor hypothesis. This is the second part of your unit 1. So let's start. So it's a continuity in the quantitative characteristics that we have discussed. So some repetition of the same from the past. So multiple factor, it is a quite natural that same small difference exists among individuals of similar genotypes due to the environmentals on the genotype. So quantitative characteristics are uh, the one with the having a show continuous variations and governed by large number of genes called multiple genes or multiple factors and polymeric genes. So that's why the multiple factor hypothesis came along with the quantitative traits. So many factors, many genes are playing role in them. So the inheritance follows the same Mendelian principles in this case, whereas the qualitative character shows discontinuous variations are governed by one or two major genes or oligogenes. So the same scientist who showed us in the previous with the kernel experiments, Nielsen LA and kernel, So in the white, he concluded these experiments with the quantitative characteristics. So he crossed red kernel, that is capital R with a true breeding of white, small r, and F1 was all medium red. Yeah, that's what we saw on that graph in the previous file. And the F2 segregated were like red into white. However, careful examination indicated the variation in the red color among the red color progenies. So F1 red was not as intense as one of the parents, and in F2 we should have observed two grades of colors. That is, one was red as that of the parents, and two were the higher red as that of the F1 individuals. In some crosses, the ratio of 15:1 was also found out, indicating that the two pairs genes were red color, that either both of these can produce red kernels. So finally, he observed different shades of red in F2 for red kernel types, and F2 showed red as white, follows as white. So this is how the cross looks like. So you have red wheat, white wheat. Yeah, so you made a cross, and then after the cross gametes, you have F1 as medium color with R1, capital R1, small r1, capital R2, small r2. Then you take the gametes, cross the, and you do a Punnett square again, and you have again like this graph. So it was concluded that two duplicate dominant genes R1 and R2, they cumulatively, they togetherly decide the intensity of the red color. So if they are present in large amount, so mainly they are all red. If they are present, one less is present, then there is a dark red, and if it is two are present, then it is a medium red. If single is present, then it is a single red. So majorly, multiple factor hypothesis says majorly the two genes cumulatively deciding the intensity of a color, and both R1 and R2 are completely dominant over the white color. And the high intensity of red color depends upon the number. How many numbers are present, that will interfere with the color of your uh, final degree. So this was not shown before in the previous graph, but this in this theory, with the help of multiple factor hypothesis, they cleared that theory. But why we are seeing these particular ratios, and it was happening because of presence of the dominant genes, and they are playing a major role in that case. Yeah, so this is again showing to you genotypic ratio and phenotypic ratio of all the genes, so making it to again total 16, the same thing. So hence in multiple factor hypothesis, so if the two parents have two gene segregations, so their cross, after the cross was done, so 1:4:6:4:1 was provided for the both R1 and R2, and which contribute to the color of a gene. So if the three genes are involved in the F2 segregations, they showed 1:, if three genes are present, then we will see this kind of ratio, that is 1:6:15:20:15:16:1 for red shades, and for 1 for white shades. So like this, if the three genes are present. So Nelson LA multiple factor states that for a given quantitative trait, there should be several genes, like in our previous case R1 and R2, which independently in their segregations but have cumulative effect on the phenotype. They segregates independently R1 and R2, but when the effect has to be there, they come together and they give the effects. And dominance is usually incomplete, so there is not a complete dominance, so there is dominance is incomplete in this case, and each genes contribute something to strength of the expression of characteristics where it's present dominance genes. These are the main factors that came exist from the multiple factor hypothesis. So what are the causes that we see these genetic variations? We see various changes in our things. So we used, so genetics, we saw thought that genetics is playing a role for like tall versus short P in the Mendel experiment. So we see always tall and short. So that might be the genetics of discrete plant phenotype might be the reason. And also the variations at single gene contributions to major phenotypic differences. So many traits of interest, often the large number of genes, each of modest effects, collectively contribute to the genetic variation. So this is multiple factor hypothesis. So from these results, there were two conclusions that we came out from the multiple factor hypothesis, majorly two conclusions. That is, sexual reproduction can produce huge diversity in phenotypes. If on the locus of two alleles, a capital and small a can produce three genotypes, capital A, capital P, capital A, small a, small a, small a, 10 diallelic loci can produce. So 310 to 6,000, 60,000 genotypes. Then second, given this huge potential diversity of genotypes, apparently new types appearing within the population may be the results of rare segregants rather than the new mutations. And so typically from these things that we have discussed, so the last thing that I want to discuss is with the an example where we are seeing the distribution of ear size, the cone. These are the cone sizes of P1 parent. So this is P1 parent with the length of 5 to 8 centimeter, and there is a P2 parents of the length of 13 to 21 centimeters. And we made a cross between them, and the F1 generation has to be coming somewhere in between, they discovered. So they were between from 9 to 15 centimeter. And further, when they crossed the F2 generation, the F1 generation, what they saw that the F2 generation seems to have all generation from 7 to 19 centimeter in upcoming increasing. So that is also one of the reason that maybe the dominant genes and the recessive genes, they are playing role having these characteristics in the F2 generations. That's what we are seeing, the diversity that we are seeing in this case. So that was about multiple factor hypothesis and qualitative and quantitative traits. So I will share these two topics with you now. What is the travel effect as it is written in the number 2 point of the characteristics of quantitative traits? Please explain the whole line. But where you are saying in the travel effect. If substitution of allele occurs in a gene's locus, if a substitution of a gene locus, then such allelic substitutions have trivial effects. Yeah, they will not have much of the greater extent of effects on the things that you're looking for. So they will be somehow less. So So here we can take a 5-minute break to start the next topic with fresh start. So it's almost 45. So we meet at 50. Yeah. So we meet at 1850 and then we start with the next topic of cytoplasmic inheritance. Okay. We just meet, just take a break and then we meet shortly. So welcome students. So maybe this will be the last topic that I will share with you today. We start with the mutations tomorrow. Yeah, so that so all are present. Yeah, everything is going fine. Cytoplasmic itself, it's saying the inheritance that is happening not because of the genetic alleles or DNA, something that is happening because of the genetic material present in the cytoplasm, all the cytoplasmic genetics, DNA present in the cytoplasm, they are being inherited to the offspring, to the next generations. That part is known as cytoplasmic inheritance. To study about that is known as cytoplasmic inheritance. So this is the second topic from today. So we are done with the qualitative, quantitative traits, multiple factor hypothesis. Now we are attaining the next topic, that is cytoplasmic inheritance. So inheritance of most of the characteristics in the eukaryotic organisms show the following characteristics features. So the contribution by both male and female parents are equal, so that the results from the reciprocal cross are identical. Segregation produces the characteristics ratio of 3:1 in the F2 generations of the monohybrid cross, and typically 9:3:3:1 in the dihybrid crosses. So this is mainly that we saw inheritance in the eukaryotic organisms in most of the time. So these features were first demonstrated by the Mendelian characteristics, such as inheritance pattern is referred to as Mendelian inheritance. It is universally accepted that genes showing Mendelian inheritance are located in the chromosomes of eukaryotic nuclei. Therefore, Mendelian inheritance pattern is regarded as a sufficient evidence for a gene to be located in the chromosomes. Such genes are termed as nuclear genes, or more commonly known as genes. But sometimes some characters that what we were seeing in the Mendelian characters, they do not show Mendelian inheritance, and they show a non-Mendelian inheritance. Yeah, there is something thing called non-Mendelian inheritance. In such cases, following characteristics are observed. So these there is a consistent difference between results from the reciprocal crosses. Generally, only the traits from the female parent is transmitted. Mainly the female parents are transmitting the results And in most cases, there is no segregation in the F2 generation. So further segregation doesn't happen in the F2 and subsequent generation. So no changes are further seen in there. So there were some exceptions, that is non-Mendelian inheritance was happening. So they group them into three categories. That is, they are those related to the cellular structures and patterns, those produced by intercellular parasites, symbionts and viruses, and those associated with the DNA containing cell organelles, that is mitochondria and chloroplast. So in addition to these cases of non-Mendelian inheritance, some characters in several organisms exhibit a Mendelian inheritance patterns also, so which are marked by individual genotype of the maternal parents. Such cases are called as maternal effects. So the things that are happening because of the maternal issues. And some evidence are there was first given by Correns in Mirabilis Yelapa and by Bohr in the Perigonium zonale in 1908. So they give the first evidence of cytoplasmic inheritance. So mainly the character of only one of the two parents, usually the female parent, was the reason that they're transmitting to the progeny. So as a result, reciprocal cross consists differences for each characteristics, and there is a lack of segregation in the F2 and the subsequent generation. Such inheritance is called as extranuclear inheritance, or extra chromosomal inheritance, or maternal inheritance. But the genes which are governing the traits showing cytoplasmic inheritance, they are located outside the nucleus. So they were not the genes that they were dealing with the cytoplasmic inheritance. They were not present in the nucleus, but in the cytoplasm. And they are referred to as plasma genes, cytoplasmic genes, cytogenes, extranuclear genes, or extrachromosomal genes. And the sum total of genes present in the cytoplasm of cells is known as plasmon, while all the genes that are present in the plastids, they are constitutively known as plastrons. So plasmon and plastrons being genes present in the cytoplasm and plastids respectively. Then comes the reciprocal differences. So some characteristics of the cytoplasmic inheritance. So first is reciprocal differences. So whenever we make a reciprocal cross, they show marked differences of characteristics governed by plasma genes, that is the genes that are present in the cytoplasm. In most cases, these plasma genes from only one parent, generally the female, are transmitting, and phenomena is known as uniparental inheritance. So mainly the female part is transmitting these characteristics, not the male one, and that's why it's also called as uniparental single parent inheritance. There is a lack of segregation in F2 and F3 generations, do not show segregations for cytoplasmically inherited traits. So this is because F1 individual generally receive plasma genes from one parents only. So we don't see many different ratios after F1 generation, because the things that has been inherited was only from the one parent side. That's why. Then irregular segregations in the biparental inheritance. In some cases, plasma genes from the both parents are transmitted to the progeny. This is known as biparental inheritance. So if the both parents are transmitting, then it's known as, then that will be irregular segregation. Then somatic segregation, these are the plasma genes generally show somatic segregations during the mitosis, a feature of rare occurrence in the case of nuclear genes. Then association with the organelle DNA. Several plasma genes have been shown to be associated with the cytoplasmic DNA or mitochondrial DNA. Then comes nuclear transplantation. If nuclear transplantation reveals as a trait to be governed by the genotype of cytoplasm and not by the nucleus, cytoplasmic inheritance of the trait is strongly indicated. And further, transfer of nuclear genomes through backcross. The nucleus of variety of species may be transferred to the cytoplasm of another species through the repeating the backcross of the former one, which is used as recurrent parent, male parents. Mutagenesis. Some mutagens like ethidium bromide are highly specific mutagens for plasma genes, while nuclear genes are not affected by them. So they are also coming up into these factors. Lack of chromosomal locations, lack of association with the parasites or symbionts or viruses are one of the different types of the characteristics of the cytoplasmic inheritance. So the known cases of true cytoplasmic inheritance are concerned with the chloroplast. So mainly in the cytoplasmic inheritance, they are in the basic in the plants. They are related to the chloroplast or mitochondrial traits, which are usually associated with the DNA. So such cases are known as organelle inheritance, plastid inheritance, and mitochondrial inheritance. If they are dealing with the chloroplast or mitochondrial traits, they are called as plastid inheritance or mitochondrial inheritance. So plastid inheritance, what is plastid inheritance is actually, so plastid inheritance is something to deal with the plasma genes present in the plastids, and that inheritance is called as plastid inheritance, and it was given by first by Correns and Bauer in 1908. And they discovered variegation, that is presence of white or yellow spots of variable size on the green background of leaves. So you see these characteristics of white and yellow color variable size on the leaf plant, and this is known as variegation. And variegation may be produced by some environmental factors, some nuclear genes, and in some cases plasma genes. So plasma genes are also playing big role in these variations. So based on that, they did inheritance of plastids in Meropolis Jalapa. The inheritance of plastids in 4 o'clock plant was first discovered by Correns. So in Meropolis Jalapa, some of the branches may have normal green leaves, while in some plants, some other branches may have only pale green or white leaves, and still others may have variegated leaves. So flowers on branches with normal green leaves produces seeds that grow in the normal green leaf. So this is what you're seeing is, so this is a type of branches from which flowers are chosen for. So there's a green, variegated green, and pale green, and this is a type of branch from which pollen was obtained. So in this case, the pollen was obtained, and this is from the flowers were chosen for pollination. And then type of leaf grown from the seed. So after the crossing over, what things that they saw. So when they grow green, they see only green. But when they grow with the pollinated green and variegated pollens, so they see only green in this case also. But variegated green, when crossed with the green, they see green, variegated or pale. They see all the three characteristics, and same hold for everyone. But in the case of pale green, they see when they're crossing with them, so they see green and pale green. So in the case of variegated green, why was that was happening and not in other cases, that we are able to see all the facts in the in our seeds, is because of the plastid inheritance, that is cytoplasmic inheritance. It is clear that variegation is determined by the genesis transmitted to the female, and that is not influenced by the type of pollen used. So mainly the female flowers that are being playing role in that. So we were seeing these results. Some more examples: maternal inheritance of IOJAP gene in the maize. So here you can see the egg regularly contribute from much cytoplasm to the next generations that does not have sperms. So what you see is they have IOJAP gene IJ small j at the chromosome 7, which controlling the plastid inheritance in the plants, and gene IJ is responsible for the normal green color of the plant further. And when normal green plant are used as a female, like in this case, I capitalize small j, and we use as female and pollinated by the pollen from the stripped with small I and small j, like this in this case here. So F1 plants are wholly green. F1 plants, you have your F1 to be mostly to be present as green. Then further, they produce green. When you make a further cross, then you have green, white, striped. Then further cross between striped and green gives you green, white, striped, green, white, striped. So mainly the female characteristics, whatever the female characteristics are present, so they were being transferred to the next generations. That is an example of plastid inheritance again, or cytoplasmic inheritance. Further, in case of male sterility in maize, pollen grain of such male steriles are aborted, and this male sterility is transmitted only through the female and never by the pollen. It was later recognized by the cytoplasmic male sterility in the maize results from the alteration in the hereditary units in the mitochondrial DNA. So inheritance of kappa particles in paramecium. So this is another example of cytoplasmic inheritance. So in paramecium, there are two strains are present. That is, one is called killer strain, another which is has a toxic substance known as paramecium. Another other strain known as a sensitive, and if it comes in contact with the paramecium, and it is getting killed if it is in contact with the paramecium, that is the killer one. So there are two types of strains, killer and sensitive. So in the cytoplasm of the killer strain, the kappa particles are playing the role. So kappa particles, they are the cytoplasmic DNA that are present, and kappa particles are absent in the sensitive strains. So they are not present in the sensitive strains anymore. So transmission of particle is through the cytoplasm, but maintenance of kappa particles and production of paramecium is controlled by the gene K, and we assume that killer strains that they're having the dominant gene, that capital K, and sensitive ones have the small k. So on the conjugations of exchange of the nuclear material, which is crossed by the small k when conjugation is for the normal time, then only the nuclear material is exchanged. Therefore, killer will produce killer daughters and sensitive will produce sensitive daughters. So here you can see the crossing of sensitive, killer and sensitive. Yeah. So there is a small k, capital A, capital K. So these are the kappa particles that they are showing. Killer genes have these present as a kappa particles in them. So when you made a cross between sensitive and between them each other, so you have both genes present. So then further delayed separation. So what you see is that inheritance of kappa particles in the paramecium. So we have many generations which have kappa particles, and few generations doesn't have kappa particles. So some are immediately separated, we can see the results. Some are taking some delaying in separations, then we are seeing the results. That's how the results are being shown. So mitochondria can self-replicate and represents another genetic system in the cell. Of course, the amount of mitochondrial DNA is so small, representing less than 1% of the nuclear DNA, is mammalian cell, and it can code for part of the protein in the mitochondria. The synthesis of cytochrome found in mitochondria, for example, is known to be present in the minute amount in cytoplasm under the control of nuclear genes, and therefore it is suggested that both mitochondria and chloroplast seems to have semi-autonomous existence, and the DNA forms the basis of uh, system separate from the nucleus. Then there is episomes in bacteria. So some hereditary materials have been found to be exist in two states, either in the autonomous states in the cytoplasms, or they are present in the or they replicate independently of the chromosomes, or in the integrated states incorporated into the chromosomes. And particle of such properties is known as episomes, including such things as sex factors. So these are episomes are mainly known as sex factors, and they apparently are not so essential to the life of bacteria because they may or may not be present in them. So they are also called as cytoplasmic factors. So what are the significance of cytoplasmic inheritance? We can understand the role of mitochondria. We can develop cytoplasmic male sterility plants like maize, pearl millet, sorghum, cotton. Mutation of chloroplast DNA, mitochondrial DNA leads to the generation of new variations. So students, I think we'll stop here today. It will be too much for you all to digest things. So we did 3 topics today. So today's topic was quite complicated, but I tried to make it as simple as possible. But anyhow, please go through this lecture one more time so that things could be easily understandable for the later part. If you want to join on my LinkedIn thing, you can send me a request. My LinkedIn name is seen with this picture, Simranjit Singh, with this degree things written there. So please be part of a LinkedIn family. So whosoever is not on the LinkedIn also, please join in LinkedIn. It's a great opportunity to be know about the various fields, what people are doing, get connected with the people in the HR department of various industries, or some HR or universities people from where you want to study, to get in contact with the students also from the university you want to study further. So this will really help you to be in touch with. So today mainly we studied about multiple factor hypothesis, qualitative and quantitative traits, and cytoplasmic inheritance. So these three topics we have governed, and we have discussed about various significances of their, what are the main qualities that we are looking for, various examples we have discussed, and yeah. So students, any questions so far, please raise your hands and you can ask now, please. Okay, good. If no questions, I would like to make it off for today. Then we see us tomorrow, same time, same place, and we continue with the mutations part of yours. Because if I go really fast with you, it will be very, yeah, too much for you then. So we go slowly and slowly and wins the race. Because you have in some days we have very less things to do, so I want to make a balance for each day so that we can each day we have good things to learn about. Okay then students, goodbye, wish you all nice evening, take care, and have a nice time, stay safe, and take care of your near and dear ones, work together. Bye bye.